Hello friends, welcome to all. I am Mr. Anil Kumar from Knowledgignita YouTube channel. Today in this lecture we will discuss about the pathogenesis of cancer, detection method of the cancer and treatment method of the cancer. In the first part of the video I completed the definition of cancer, sign of cancer, types and etiology of the cancer. If you missed the first part of the video, go through the link present in the description below. So let's start the pathogenesis of the cancer. Here we discuss the different types of mechanism that behind the cancer occur, and it is a very important part of the cancer and also exam point of view, so that listen carefully. First you can see the diagram of normal cell cycle. In normal cell cycle, the whole cell cycle process is completed in a four phases, that is G1 phase, S phase, G2 phase and M phase. And and three checkpoints are there in cell cycle, and these checkpoints are control the vital function of the cell cycle. The first one is G1 checkpoint. The main work of G1 checkpoint is to check the DNA and different cellular machinery like growth factor, nutrients are damaged or not. Only repairable DNA are passes to S phase for synthesis, and unrepairable DNA and different growth factor are commit to suicide. Then the second is G2 checkpoint. These checkpoints ensure all the chromosomes have been replicated and that the replicated DNA is not damaged before cell enter to mitosis. Then the third one is metaphase checkpoint. In metaphase checkpoint, these checkpoints generally determine whether all the sister chromatids are correctly attached to the spindle microtubule or not, or before the cell entered the anaphase stage. Due to these checkpoints, the whole cell cycle process is controlled condition. But these checkpoints are damaged. But these checkpoints are damaged due to some mutagenic agents and carcinogen. Due to mutation occur in cell cycle, the production of abnormal cell takes place, and that leads to cancer. The most common example is food colorants. Generally, food colorants are used for the attractiveness of food, especially foods like chicken pakoda, KFC, and food prepared in hotel and restaurant. The carcinogen present in food colorants is benzidine, 4-amino-azobenzene, preservative like sodium nitrate. This chemical contain food colorants are damage the checkpoint of the cell cycle. Due to damage of checkpoint occur, there is no regulation of cell cycle takes place, and abnormal cell formation is more, and this abnormal cell leads to cancer. This is the first pathogenesis behind the cancer and how the cell cycle produce the cancer cell. Then the second one is role of apoptosis in cancer cell and normal cell. You know very well about apoptosis. The apoptosis process is the form of programmed cell death occur in our cell. You can see the diagram. In normal cell, the cell division process is tightly regulated, and old and different damaged cells are undergo apoptosis as the new cells are produced. But in case of cancerous tumor, the cell division get out of control and do not undergo normal apoptosis, so that the continuously proliferation of the cancer cell is occur, so that tumor formation or different cancer are produced. Then the third one is activation of proto-oncogene to oncogene. Proto-oncogenes are the genes that normally control the cell division, apoptosis, and differentiation of the cell cycle. Generally, proto-oncogenes are not cancerous. At present, they are not harmful. After mutation takes place by some cancer-causing agents, these proto-oncogenes are produced and activate the oncogene. And these oncogenes are highly dangerous and cancerous. And due to the oncogenes having the cancer-like properties, highly cancer-like property, they release the cancer cell. Then the fourth pathogenesis is inactivation of tumor suppressor gene. The tumor suppressor genes are suppress the cancer producing cell. The most common example of a tumor suppressor gene is p53, PRB, or also known as protein retinoblastoma. Generally, these genes present in the G1 phase of the cell cycle. Whenever the cells are mutated, the normal tumor suppressor genes are repair the mutated cell and signal to activate the apoptosis process on unrepairable cell. But in defective tumor suppressor gene are not able to repair the cell and signal to the apoptosis process, so that inactivation of or defective tumor suppressor genes that can cause cancer. In this slide, we are discussing about different detection method of the cancer. Here, there are different detection methods are there, and we are discuss one by one. The first one is biopsy. In biopsy, the biopsy means the removal of tissue from the tumor region, and that tissue is examined under the electron microscope and identify the abnormality. And by using this test, you have to determine the cancer, detect the cancer, and it is a completely a histopathological test. Then the next one is blood test. Already you know, in leukemia, tumor are not formed due to enormous number of WBC in blood. The cancer is leukemia is occur, so that blood test is used for the detection of the leukemia or blood cancer. Then the third one is PSA, prostate specific antigen. Generally, this test used to detect the prostate cancer, and this PSA test is especially in the initial stage of prostate cancer. Initial stage of prostate cancer is detected by the PSA. Then the fourth one is mammogram. The mammogram, specially this mammogram, is used to detect the breast cancer. Then the fifth one is CT scan. In CT scan, here CT means computerized tomography. With the help of some ionizing radiation and computer, we can take the 3D image of the particular organ and tumor and detect the cancer. Then the MRI. In MRI, it is the most popular method nowadays, and in the MRI means the magnetic resonance imaging. With the help of magnetic field, we have to detect the cancer, and it is a very safe technique to detect the cancer, because in this technique radiation are not used. Then the next one is pap smear test. Generally, this test is used to detect the cervical cancer. Generally, the human papilloma virus causes cervical cancer, and the cervical cancer is detected by the or diagnosed by the pap smear test. Then the position emission tomography. Generally, this test is used to detect the different types of cancer like intestine, large intestine cancer, lungs cancer, breast cancer, and different types of monoclonal antibody also used to detect the cancer. Now, in this slide, we are discuss about the different treatment method of the cancer. Before I start, it is special announcement for you. We conducted the free MCQ related to this lecture. After completion of the video, go through the link present in the description below, and if you attempt the test, I feel better for this lecture. So let's start the treatment of the cancer. There are different types of treatment methods are used for the cancer. First one is surgically removal. For example, if the tumor occur in uterus, by using surgically method, the uterus is removed. Then the second one is radiation treatment. There are some ionizing radiation like X-ray, gamma ray, and UV ray. By using this radiation, the cancer cells are killed, and treatment of cancer is occur by some radiation treatment. Then the chemotherapy. Some chemicals or some chemical agents, some formulated chemical agents are used to treatment of the cancer, and generally these chemotherapeutic drugs are highly side effects are more. Then some anti-cancer drugs or some immunotherapeutic drugs also used to treatment of the cancer. Then some biological response modifier like alpha interferons, monoclonal antibodies, and gene therapy. And this is one of the treatment method of the cancer that used for the diagnosis and prevent the cancer. In the next video, we are discuss about the pharmacology of anti-cancer drugs. So it is the most important part of the cancer, and what are the different types of drugs are used for the treatment of the cancer. So I'm making them this video separately. Thank you.
Thank you for watching this video. If you like this video, go to the like button and click it. If you want to share, you can. And if you are not subscribing our channel yet, for what you are waiting for, man, go to the subscribe button and click it. क्योंकि सब्सक्राइबर तो मांगता है ना यार.